Okay, good morning, everyone. Thank you for showing up early. The poster time was right, thank goodness. We are in the large lecture room where we don't normally hold our defense seminars, but this is a pretty major milestone, not only for Rafael, who will be presenting his dissertation today, but also for the Cameron Lab. After 5 years, the first of our PhD students is ready to leave the nest. And I really couldn't be more happy and more proud of Rafael to be the first. As a good systematist, he has demonstrated competency in the field, as you see, in the herbarium through nomenclature and taxonomic revision, in the molecular laboratory and phylogenetics, in the morphological laboratory as well, and also in the area of chemical ecology. So bringing all of these different subject areas and tools together to address a fundamental question in orchid systematics focused primarily on pollination biology. I also look out into the audience and see people from entomology, from the Forest Products Research Laboratory, from downstairs, from upstairs, from Columbia, and everywhere in between, which I think also epitomizes Rafael's research over the last 5 years being multidisciplinary and quite intricate. Before I hand the podium over to him, I want to just give you a little bit of background. Rafael completed his bachelor's and his master's degree from the Universidad de los Andes in Bogota, Colombia. And we actually met, 8 or 9 years ago. When he was a master's student, he came to New York and worked in my lab as a Latin American fellow sponsored by the Mellon Foundation. And then when I came here to Wisconsin and he was looking for a PhD program, he considered two places, the University of Florida and the University of Wisconsin. Unfortunately, our recruiting season happens in February. So he spent a week in Gainesville. It was nice and sunny and warm and pleasant. And then he came to Wisconsin. And I remember the day I took him out onto Lake Mendota, and I said, go ahead, you can walk on it. Go on. He's like, really? And, yeah, you can walk on it. Well, I was sure he wouldn't come after that freezing cold week, but I'm happy that he did. And he has really excelled in so many different areas. He was supported initially as a project assistant in the herbarium, moved on to become a research assistant, partially funded in the Forest Products Lab. He served as a teaching assistant here in our department for several semesters, ultimately winning the College of Letters and Sciences Outstanding Teaching Fellow, one of our most prestigious awards. He also then was supported on an Allen Fellowship in the last year. In addition to those financial fellowships, he was named a Kohler Scholar, which allowed him to live on campus in the Knapp House. Some of you know about that, which is also quite competitive and quite an honor. He's already published several papers, including 2 from his dissertation and 1 that's in press from his dissertation. I also noticed an odd publication here just this year on genetic structure and molecular variability of cucumber mosaic virus. So he's branching out, apparently. He has succeeded in funding his research through many different avenues, including a National Science Foundation Doctoral Dissertation Improvement Grant. I tallied all this up, I'm going to embarrass him. He brought in $29,500 in the last 5 years to support his research, and that's not considering assistantships and fellowships and that kind of thing. So he obviously has a bright career ahead of him. He's already being courted by orchid societies as an invited speaker. He did a tour of Portland, Oregon, Eugene, Oregon, Corvallis, and Seattle, talking to local orchid societies. He was an invited speaker at the Monocot meeting in New York last year, and also gave papers at the botany meetings in St. Louis, Columbus, and an orchid conference in Guayaquil, Ecuador. So I think those of us who are faculty know that teaching, research, and service are 3 of the big areas that we look for in our colleagues, but also that we look for in our future colleagues and our graduate students. And I think Rafael has certainly excelled in all of those. and I hope that you will join me in welcoming him as he presents his dissertation defense on the phylogeny, floral micro-morphology, rewards, and fragrances in the neotropical orchid genus Mormolyca of the maxillary. Rafael.
So I want to start by thanking you all for being here. It is a pleasure for me to share with you the research that I've been doing the last 5-6 years here at the department. So I would like to start first with my favorite evolutionary biologist, and I hope that yours, Charles Darwin, and his book on the fertilization of orchids by insects. He later called this a flank movement on the enemy of his theory of natural selection. Because basically in this book, he goes ahead and describes in detail a bunch of mainly European orchids and how their morphology is related to their pollination or to their pollinator. In it, he basically treats a couple of European orchids and some examples from the New World. He's amazed by this catasetum, where the pollen-bearing flower differs drastically from the ovule-bearing flower. And then he also mentions these Coryanthes and Cynopes, and how this bizarre flower morphology has to be the result of coevolution with their pollinators. A very famous account in this book is this orchid, Androecum sesquipedale, from Madagascar. He receives this orchid, and based on gross morphology, so the color, the white color, this long spur, he predicts that the pollinator has to be a moth with a tongue long enough to reach the nectar here at the end of the spur. Unfortunately, he died, and that pollinator was never discovered, but years later, the pollinator actually was found, and just as he predicted, it was a moth with a proboscis or tongue long enough to reach that nectar. So following his example, I'm going to use also morphology, observations, and experiments to describe or predict the pollination biology of a group of orchids found in the neotropics. The group is named Mormolyca, and I'm going to start by using or reconstructing the evolutionary relationships within the group, then look at its morphology, and then study the floral scent variation of some species of the group. So Mormolyca in this tree that shows the relationships within the orchid family is found in the biggest subfamily of the orchids, the Epidendroideae. Within this subfamily is found in the tribe Cymbidae, and within the tribe Cymbidae in the subtribe Maxillarinae. And Maxillarinae are a very diverse and conspicuous groups of orchids in the New World, tropics. So if you walk into a forest in the New World, you will certainly find a bunch of these species. Now, relationships within the group were unclear until relatively recently, when Mark Whitten and Mario Blanco from the University of Florida, based on DNA data, reconstructed the relationships within the group and came up with 17 well-supported clades. Now, most of the species originally were named under this genus, Maxillaria, and this went from 500 to now 250 species. Some genera had to be created, others changed drastically. That is the case of Mormolyca, because it originally included 6 species. After this revision, it now includes around 25 species. And why did this happen? So when we looked at their reconstruction, we see here the Mormolyca in its original sense as paraphyletic So all the descendants from the most recent common ancestor and are not included, related to a big clade that includes species of Maxillaria that were all included in a section known as fruit specimens. Embedded within, we have here the genus Chrysocygnis that only includes 2 species. So to achieve monophyly or to use a name that included all the descendants of the most common recent ancestor, they decided to change this species into Mormolyca. Now, this is just an example of what Mormolyca looks now. So we have here species of Mormolyca that were previously recognized within the section Professors. And this morphology here is what used to be taken to call them Maxillaria, because this is the Maxillaria flower type. Now, this are the flowers of the Mormolyca in its original sense. This, what we're seeing here, could be an example of how pollinators are actually directing the evolution of this group, and this would be just the result of convergence into a flower type, a previous flower type or another flower type, the Maxillaria flower type. So let's see, how do we recognize Mormolyca now? So first of all, I want to start by saying that this, the genus was originally described by the Austrian botanist Edward Fensel in 1850, based on this specimen. The name Mormolyca comes from the word mormos, that means hobgoblin, and apparently it alludes to the bizarre coloration pattern that the type specimen flower has. They are found all the way from southern Mexico to Peru and Bolivia and northwestern Brazil, including the Greater Antilles. They are recognized by this unifolia, suitable, always subtended by a pair of papery bracts, and the single flowering fluorescence always coming from all parts of the rhizome, rather than from the apical growth, that this occurs just in this group. So this combination of characters allows us to know that this is a Mormolyca when you see a live plant and when you see a plant in the herbarium. So, okay, so another really interesting thing about this group is that Mormolyca ringkins is maybe one of the 2 species that has been studied, orchid species that has been studied in detail its pollination. And Rodrigo Singer was able to see how, or they were able to determine that the labellum here of the flower has volatiles and non-volatiles that mimic the pheromones of the male of its pollinator, a bee of Scaptotrigona. So the male falls for the trick, thinking that this is a female, and while trying to copulate with the flower, the pollen gets attached to its back. Now the male obviously has to be young and naive to fall again for this trick and be able to transfer the pollen to another flower. So in addition to this, so this pollination system is called sexual deceit, and it's basically based on deception, right? Another type of pollination that is thought occurs in this group is based on rewards. So bees have been observed gathering the trichomes that are found here in the lips of the flower. They might harvest these trichomes for as a reward for food, or there are species that have these resin-like secretions on the lip. Bees are known to harvest these secretions either as a food reward or to use those resins in the constructions of their nests. So this could be also another type of reward that occurs in the group. So this is a really interesting thing that's happening here. 2 pollination systems deception and reward occurring in a really small group of orchids here in the New World. So, how can we explain this floral diversity? It's very possible that it's due to its pollinators. So, what I'm going to do, again, is I'm going to, just as Darwin did, I'm going to gather up evidence from morphology, from the variation in the floral sense, and I'm going to use the evolutionary relationships of the group as a framework to study the evolution of these characters. See if I can gather enough data to predict the pollination biology of the group. So I'm going to start with the molecular phylogeny. So what we did basically was work upon Mark's written original work. We increased taxon sampling. These stars are just showing one or more individuals and the areas where they were collected. There is a big gap here. However, we think that this is just one species that has a wide distribution. And that species has these Mormolyca regions and has been collected here in the northwestern Brazil. We included also 38 taxa that represented the different grades of Maxillarinia. And in total, I was able to get a hold of 93 accessions representing 82% of the species found in the genera, in the genes. So, I used the same markers that were previously used for the study. So, ATPP RVCL, MATK, TRLK, RPOC1, these are all chloroplast markers. And then a new marker that I implemented was the 3' portion of the YCS1. Also, in addition to the internal transcribed spacer of the ribosome, I included the external transcribed spacer, and I experimented with the low copy nuclear DNA gene 5C. And what you're seeing here is just a bird's eye view of the matrix that includes all the sequences that I analyzed. So I want you to remember this gene, these chloroplast markers, RPOC1, how long it is, and then on the other side, this external transcribed spacer of the ribosome, how short it is. And I'll mention them again soon. So I use this data to reconstruct the relationships within the group. Parsimony had been used originally. I went ahead and used parsimony, and also I did a Bayesian approach, basically a probabilistic approach. It gives you the posterior probability of the tree given the data. So remember, so this is what the sequences, the information that the sequences have. So remember that long chloroplast gene, RPOC1? Well, actually that was the least variable, or it had the least amount of variable characters. Now that ETS from the ribosome, that it was the shortest one, well, it was actually had the greater amount, the greater amount of variable characters, or of informative characters. So I went ahead and studied and analyzed individually each of these genes. I did a combined analysis of the chloroplast genes, of the ribosomal, markers, of the ribosomal markers, and the 5C, low copy nuclear genes. Looked at the topology and found that there were not major differences in the topology. So I went ahead and combined all this data and did a total DNA analysis. What I'm going to show you are the results of this total DNA analysis. This is what it looks like. So basically, we've strengthened and validated previous results by the researchers at UF. We've also found higher resolution, not only among the genera, but within Mormolyca. And something really cool was that during my research, I collected species that was unable to identify initially. So the first thing that came up was that they could be new species. So I went ahead and added them, sequenced them and added them to my analysis to see if those species actually came up where I thought they would be. So Actually, I assigned them to a genus and then confirmed that assignment using DNA. So we found 2 species of Maxillaria that were found within the other species of Maxillaria. 1 species of Ornithidium that also was found with the rest of the Ornithidium species. Found 3 species of Mormolyca, 1 from Colombia, another one from Ecuador, and this species was, as soon as I saw it, growing at the Mary Selby Botanical Gardens, based on those characters that I previously talked about, I knew that it was a Mormolyca. Had never been described, appeared right as I thought, within the genus Mormolyca, but unfortunately, there is no label for this plant, so we don't know where it comes exactly. So, nonetheless, we went ahead and described this new species. The 2 species of Maxillaria and the Ornithidium were described with the name Eric Christensen. An undergrad student from here, Daniel Bergquist, and I described the species from Colombia. The Mexican botanist Germán Carnevali, who collected the species in Ecuador. We went ahead and described the species with him, and then with Ken, where we described the new species found at Selby Botanical Garden. Okay, so let's look now at the phylogeny of Mormolyca. Again, we see this grade of species that don't share, or that not all the descendants of the common ancestor are included, but they have this common characteristic that it's the insect-like labellum. They all share that insect-like labellum. No perceivable scent, at least for the human nose. They are related to a big, well-supported plate. That plate is divided into 2 subplates. This one is characterized that most of the species have by this glossy labellum. And they tend to have faint, sweet fragrances, except this guy here, Mormolyca pureblica, that has a smell that resembles overripe banana. This, the only species that doesn't look like the rest here is actually Mormolyca dresleriana. This species shares a character that is shared by this subclade, and that is that the labellum has a linear path here in the medial region. All the species from this clade have that linear path in the labellum, and most of them actually have strong, sweet fragrances, a mix of vanilla and chocolate. Okay, so in summary, we have again this species here with the insect-like labellum, scentless. I am hypothesizing that all these species are actually sexually deceptive, and I'm going to call this group for now the ringheads group from now on. The species of the glossy labellum and the faint aroma, I hypothesize that those species should be offering some type of food reward related to that glossy labellum. I'm going to call them, I call this group the heteroichae. And the species with the linear path, I also hypothesize that this is food reward, and I'm calling this group the rufescens. So, and this group actually includes the Mormolyca dresleriana. Okay, so we've reconstructed now a phylogeny, and I'm going to use this framework to look at the next step. First, the macro and micro-morphological characters, in addition to some histochemistry analysis. So I went ahead and sampled fresh material from this species here. I looked at specifically at the labellum, hand sectioned the labellum, and looked at it in the microscope. I was interested in defining or seeing what type of structures are found here on the surface of the lid, and also see if there was some difference or some characteristics in the tissue that makes up the lid. Once I gathered that data, I also did some histochemical essays. So I used Lugol solution to look for the presence of starch. Starch grains stained black or black-blue. I used Sudan 3 and 4 to test for the presence of lipids. Lipid bodies stained red, and then I used a xanthoproteic test to look for the presence of protein, specifically aromatic amino acids. These amino acids are known to be produced exclusively by plants, not by animals, so this could be taken as a reward. I went ahead and then analyzed the presence and absence of all these characters, including the presence of this nutrient, using statistical and phylogenetic methods. So these are just this is just showing the diversity of papillae or structures that I found on the surface of the lid. These drawings were done by an undergrad, Galen. So what we have here are the capillary or the hair-shaped papillae, clave, conical, fusiform, globose, long cylindrical, ovoid. These piriform are actually like a hair-shaped papillae that you can see here, and the piriform and spatulate papillae. So let's see how they are distributed. In the ringhands group, the flowers with the insect-like labellum, we found that most of the species have capillary papillae. And that capillary papillae tends to be concentrated towards the margins of the lid. Most of the species tested positive for the presence of lipids in those papillae, and just one species tested positive for starch. However, that starch was not found in the papilla per se, it was found in the tissue underneath, usually the papilla. And I'll talk about that in a while. In the Hedwige group, the species with the glossy labellum, we found that most of the species have this long cylindrical papillae. In this case, all of them tested positive for lipids, so even that secretion that makes up the glossiness of the lid tested positive for lipids. And most of them tested also positive for proteins, except Mormolyca fulerica, the species with the smell that resembles overripe banana. Again, we found a species where starch was present, not in the papillae per se, but in the tissue underneath. And this is what I want you to look for. So you see how in this section, the tissue here, the cells are very different from the cells underneath the papillae. These cells tend to be smaller, have a dense cytoplasm, and usually they have bigger nuclei. This is characteristic of glandular tissue that occurs in other orchids and is known as osmophores or scent glands. So apparently this tissue in other orchids is known to synthesize the volatiles that give the aroma to the flowers. Okay. The rufescens group, the species with the linear pad on the labellum, they all presented papillae, like clave or spatulate, that usually have a distended apex, so like the clave or spatulate, like this one. They all tested, most of them tested positive for starch, lipids, and protein. The papillae, they all tested positive for that. Okay, so when we looked at the relationship of the presence or absence of these characters in the species, we can analyze that presence or absence using statistical methods. So this is just display of a correspondence analysis. The arrows here are just showing the contribution of each of the characters or traits, in this case the papillae, to the groupings of the species. The intensity or of the color also indicates the intensity of the contribution. So, for example, here, capillary papillae are more related to these species that are the species from the ring-hens group. The spatulate papilla is more associated to species here that belong to the rufescens group. And there is a weak relationship here between fusiform papillae and these two species, with Ricci and the Guigia. Now, how do this look in the phylogeny, the presence or absence of these characters? Well, the capillary, again, look, absent in these big plates that includes the rufescens and the Guigia group, just present in the ringhead. The fusiform papilla, remember that they're just present in Hedwiga and Ricci. Well, it's not a coincidence that they are actually sister species. And then the spatulate papilla are just present in some species of the rufescens. Other papillae were present only in most of the species of these two plates and not in the ring. Now, in relation to the presence and absence of these nutrients, proteins, again, in these two plates present, not in the ring-hens group. The same with starch, but starch is actually just present in those species that have the ligure pad. This is just showing you or reminding me that this is just starch that was present in the tissue underneath the papilla, not in the papilla per se. And lipids, again, present throughout. Okay, so I studied how these characters could have evolved in the phylogeny. And what I'm going to show you here is just a stochastic character mapping where red represents the presence of the state, blue the absence of that character. And this is just the probability of the evolution of those traits in the phylogeny. So you see how capillary papillae are present since the origin of the groups, long cylindrical and lost in this big clade. Long cylindrical are also present since the origin of the group, but lost in this subclade, the rufescens. Fusiform just evolved in those two species, and spatulate apparently evolved multiple times only in species of the rufescens. Now, papillae like the spatulate papillae, like the ovoid or the pyriform, that are just present, and it's very probable they just evolved in this big place. When we looked at the presence of nutrients, it actually is related to the evolution also of proteins and starch. So the appearance of this papillae is also related to the appearance of this nutrient. Okay, so we looked at the micro-morphology and some micro-morphology, looked at histochemistry and see how these characters evolved using a phylogeny as the main framework. And we see how the presence of some nutrients seems to be related also to the presence of specific papillae. Now, the next step was to look at the floral scent variation. And what I did here for this part of my research, I was very lucky to be trained by one of the best chemical ecologists in the country, Dr. Robert Raguso. The good thing about that is that he made it look very easy. The bad thing about that is that he made it look too easy. So once I was here, I found out how hard and difficult this, just to do this, is. So I'm going to show you some examples. Just two species from the rufescens group, the Mormolyca rufescens and Arévalo. Two species from the Hedwigae group, Mormolyca hedwigae and Mormolyca, and two species from the ring-hens group, Mormolyca ring-hens and Mormolyca hedwigae. Now, how I collected the scents of the flower, but I dissected the flower. So the scents were collected via hexane extraction. So each of these floral parts, or the sepals and petals and labellum, were extracted separately. I used hexane and left this for 20 to 24 hours. This extraction later was filtered and it was analyzed using a gas chromatograph mass spectrometer. This instrument is found here in the department. It's a shared instrument between the horticulture and entomology department. And then I went ahead and analyzed or identified the compound. I'm going to show the results only of the labellum and column, since as I've shown, variations, small variations in the labellum seem to be related to specificity in pollination. So I'm just interested in the compounds that are mainly or solely produced by the labellum. And also in the column, since the column seems to be also related to the pollination in orchids, but you'll see why I ended up also looking at the column in a moment. So how does this GCMS work? That sample is injected here, volatilized, and transferred to the GC. This is just a temperature, this is just an oven whose temperature increases gradually. So as the temperature increases, compounds are volatilized. Small compounds volatilized faster and go through this capillary column and exit faster than the big compounds. So what you get here at the end of the analysis of the GC is this. This is the time that the run takes place, and each of these peaks represents a compound exiting the GC. So again, small compounds, bigger compounds. They exit the GC, go into the mass spec. Here, each compound is bound by electrons, and it's disintegrated. Each particle that is the result of that bonding is red, and then you have here the mass, the charge per over mass, sorry, mass over charge of each of those particles that are the result of what happens here in the mass spec. So you use these two data, the retention time peak and the mass spec, to identify the compound. Each of these peaks, again, is related to a specific compound, and the area is also related to the concentration of that compound in your sample. Okay, so this is what my samples look like. This is just an extraction of the lid. When I looked at the extraction of the column, this is what I got. So what you can see here is that the column is not only producing the same similar compounds than the lid, but it's also producing higher yields of those compounds. So it seems that the column is it's also producing volatiles or non-volatiles that could be important in the pollination of this species. Okay, so what did I found? So in most of the cases, the yields of the extractions were higher in the column than in the lid, as you can see here, except in Mormolyca peruviana. Of those extractions, of those yields, most of that extraction was the result of compounds that were exclusive to the column, or exclusively produced by the column. So the column tended to produce at a higher amount of compounds found exclusively on it as compared with the labellum. Now, when I looked at the variation of this compound, so the quantity and quality of the compound, there was no significant difference in the variation or variation within the structures of a species was no different than the variation among species. So unfortunately, I couldn't differentiate each species based on the compound or the amount and the quality or type of quality of compound. However, when we look at the type of compounds, there are things that are interesting. So the first thing here, this is just a representation of the compounds grouped by the phylogeny of the species. So dark those dark bars represent major compounds that have made up a high percentage of the concentration of the yield. The dark gray are minor compounds, and this light gray are just traces of compounds. Now, as you can see, many of the major components of the extractions have high retention time, so they could be big molecules. And also many of those abundant compounds are shared by more than 2 species. Now let's see, I identified some of those compounds. When you look at the actual identity of the compound, this is where things get interesting. So this, we found these aromatic benzenoids and this sesquiterpene in Mormolyca aureolobula. They are known to occur in the floral scent of other flowers and orchids. However, I want you to remember this methyl salicylate in Mormolyca aureolobula. I'll talk about that in a minute. Aldehydes, like nonanal and octadecanal, are known to be related to or play important roles in the pollination of food-rewarding and food-deceptive orchids, and also seem to be important in the pollination of sexually deceptive orchids. So we found this octadecanal as a major compound in Mormolyca peruviana, one of the insect-like flowers, but look how there are traces of that compound in the rest of the species. And we found nonanal only in aureolobula. Eugenol, this is a phenylpropanoid that is known to be collected by orchid bees, euglossic bees. And these compounds with methyl salicylate are collected by the bees to be used later to attract the females. So it is a type of reward. It's not a nutrient-based reward, but it's a reward that is used by these bees to for the reproduction. And again, look where it's found in Mormolyca aureolobula as a major compound, eugenol. Fatty acids, these have been found in other orchids, seem to be related to the unpleasant odor of some of these orchids. However, look, it's interesting where they were found, most of them, or 2 of them, in Mormolyca culearica. This is the species with the smell that resembles overripe banana. And also, it is known that this tetradecanoic acid that was found in Mormolyca rufescens is found in pollen collected by honeybees and seems to have important antibacterial properties that are important in the nutrition of the bees and in the maintenance of the health of the hive of the bees. Okay. And but we also found alkenes and alkenes. These are the compounds known to occur in insects and serve as pheromones. They occur, again, not exclusively in the species that I thought or are known to be sexually deceptive, but look, they occur in the rest of the species too. So, and last, the triterpene squalene was found in all of the species, and this was just interesting because this has been found in another orchid species, in the oils present in another orchid species on a New World or orchid on seed. Okay, so some of the findings that what I found up to now seem to be consistent with previous results in orchids, in Maxillarine, and basically also in Mormolyca rheniens, basically the alkanes. Though I have been, I can't positively or significantly differentiate species based on scent, there are motifs that are becoming evident, like Mormolyca aureolobula, and like the fatty acids now become. So in aureolobula, remember those eugenol and methyl salicylate that are collected by orchid bees, and in Mormolyca aureolobula, those fatty acids that could be related to the smell. Okay, so I think In conclusion, we were able to construct and also supported a hypothesis of the relationship between Mormolyca. We used this hypothesis to look at the evolution of characters. Also, we can see that the types of labella, in combination with the papillae and the presence absence of certain nutrients, is defining morphological groups. And those groups are related to the pollination system. If this is true, you see how the ringheads group here, that is the sexually deceptive, is actually the ancestral state in the whole genus. And this would be the first case in orchids where we go from a deceptive pollination to a rewarding pollination. So this would be at least the first known case in a New World group of orchids. And we found also these alkanes that are related to the pollination in sexually deceptive orchids. We found them not only in what I hypothesize are sexually deceptive, but in the rest of the orchids. So they could just be vestigial traits from these deceptive orchids that may or may not play a function in their pollination. We don't know that. And again, we also found this new, or what seems to be this new or record of compounds, at least in this group, that we hadn't found them in Maxillarine, and they could also be showing us that there is yet another type of pollination occurring in the group, not related with rewards, nutrient rewards, but rewards in the form of molecules or compounds used for the reproduction of the pollinators. Okay, so with that, I would like to acknowledge first Ken, his patience and support. It's been great. My committee, Ken Seisma, David Bao, Dan Young, Hiroshi, thank you for jumping in at the last moment. My collaborators, the late Eddie Christensen, Barbara Carlsworth from the Eastern Illinois University, Rob Raguso from Cornell University, Mark Britten and Mario Blanco from the University of Florida, Santiago Ramirez at UC Davis, Patricia Harding from the American Orchid Society. My good friend and advisor who encouragement has taken me where I am right now, Julio Betancourt in Colombia. Here in botany, all the present and past graduate students, not only from my lab, but from the Seisma lab, the Baum lab, and the M. Schroeder lab. I would like to, I've had the pleasure to work with undergraduates, so, and they've been very helpful in producing some of the data that I showed today. So Sam, Mariam, Gayden, Anna, Zach, specifically Gayden, has been very enthusiastic. He's already a co-authoring a species. He's already a co-author in one of the papers that we submitted. So it's been a pleasure to work with him. The staff, Sarah, her help has been invaluable for submissions, for talks, posters, papers, the images that we're seeing here. So Mo also, who allowed me to grow my orchids in the greenhouse, he initially just gave me a space maybe this big, and now I have like 4 times that. But we have the biggest collection of Mormolyca in the world, right? So thanks, Mo, and all the staff at the greenhouse. Mike, who always, when I was teaching, he subbed for me so that I was able to go give talks or go to the field. Thanks, Mike, for that. Ted, who's like I respect him as a taxonomist, as a systematist. His help in when I first started describing the species was also very, very useful. Other people here at UW, Ken and Johan from entomology, Beth from horticulture, she's the lab tech that is in charge of the ECMS, and Alex at the Forest Products Lab. My funding is the NSF Doctoral Education Improvement Grant, all the grants, awards, and fellowships here at UW Botany Department. The American Society of Plant Taxonomists and the Botanical Society of America for the Graduate Research Awards, Sacramento Orchid Society, the Color and Fellowship, all the institutions that gave me plants so that I could study them. My friends here in Madison, the Colombian Mafia, my friends here at Hypatia, thanks, and my parents, who came all the way from Colombia, my sister, that's been great, and my grandmother, who has been my inspiration since I was a little kid. Thanks. So with that.
Questions for Rafael? Yes?
Do you ever find the sexually deceptive fruit forming a monophyletic clade based on like different genes or maybe like nuclear versus?
No, the only weird thing that we found is here, Mormolyca polyphila is the only one that jumps when I looked at the chloroplast and the nuclear. Not always, doesn't always appear within Mormolyca. It comes as, you see how it's the sister to the rest of the species when I do a combined analysis. That's the only one that has like a weird, and I suppose it's sexually deceptive. And also that the morphology is very different from the rest. Questions? Yes?
Are all the orchids in your group pollinated by bees?
Yes, mainly that's what I suppose, that they are. Given the what I found in the scent and even the characters, I suspect that most of them are pollinated by bees. Although who knows, maybe these resins are also harvested by other types of insects. However, just like what happened with Darwin, I hope that I am able to find the pollinators of the species before I die. This has to be proven in the field. Any more questions?
I have a question. Yes. I don't think you talked a lot about the trichomes and how the insects actually use them. What is known about that in terms of how they're harvested, how they're used, and what's the occurrence in the plant kingdom of insects collecting hairs?
Yes. So in orchids, it's known to happen in maxillary. So there's just one account, a single account of a bee harvesting these trichomes, and apparently what was happening is they saw just the bee trying to. Oh, Dan, you're gonna. I forgot the name of the structure, but they were just harvesting them or eating them, not eating them, but they're collecting the trichomes in the papillum. Now, there are also other species that are known to appear that don't have other type of trichomes, and what they actually are known as pseudopollen, and bees collect them also and actually ingest them. Now the resins are known to be collected by bees, but they are not ingested, they just take them. Apparently they use them for their nests. But that's what's known of the use of these trichomes, at least in maxillary. Yes?
Thanks a lot, that was really interesting. I'm wondering about the implications of the work, and I know that you want to find these pollinators before you die, but just interested in how stable or endangered some of these orchids or their pollinators are, and if there's any risk of not being able to figure this out before they die.
There is actually a high risk, now that you mention it. So, something that I find fortunate and unfortunate is how all these institutions and nurseries now hold the diversity of orchids that was once known to occur in the wild. Actually, they've been able to collect it. Forests where these orchids occur are rare. They are, like, I've been able to find these plants in the wild, and I've had people send me pictures of collections of these plants in the wild. So they do exist out there in the wild, but in very, like, how would, like I said, it's like faraway ports, like places that it's hard to get to. So I've been able to travel throughout Colombia for this research and for my own studies, and after, I can tell you that after at least 2 days on a mule, we get to places where the forests are still in patches, you know, that people have cut the rest of the land to bring in cows. But in those forests, we've been able to find this species, and I suspect that the pollinators are there. Right? It is hard to find actually this pollination occurring in where these plants are grown in the nurseries, but I am very sure that in the wild, they should still exist.
Any other final questions?
One more item. Going from deception to rewarding, what abiotic or biotic factors out there cause that?
From deception to reward. So the idea that has been, what it's always thought to occur, what originally was, or occurs in other groups, is that they go from reward to deception, right? So it is apparently the appearance of these novel compounds that are used by pollinators, or these novel compounds that trick pollinators into thinking that there is this, that it's a female. In this case, those compounds apparently already exist, but if that is the case, if that's what's happening, and it's thought that happens in the other cases, we can see that here when we see the appearance of those other compounds like eugenol and methylsalicylate that were not present in this group but appear in some species, or should appear in some species of this group. So there is the appearance of special compounds that are now are not attracting pollinators or tricking them, but actually are rewarding, and also the appearance of trichomes like this that have now nutrients in them. So that's what I think is happening. The evolution of these structures that now then. Oh, and the other thing is that all the machinery to synthesize the fragrance, remember the glandular structure, was already here. So because osmophores were present here, producing all these long chain hydrocarbons, and now they are being used, but to synthesize other types of rewards, like starch or protein. So the machinery was already there. So it just changed to production of new structures.
Thank you.
So the committee will move on to a barbecue where we grill Rafael for the next 2 hours upstairs. Assuming that he survives the grilling, please join me in my lab up in room 222, maybe around 12 o'clock for a celebration. And with that, let's give Rafael a round of applause.